an interesting setup. He is actually a breast cancer surgeon. For the scientists and the physicians in the room, yes, surgeons can do science, okay? And he is a good example of that. He actually has a great passion for doing science, and he is now collaborating with Giselle on work that he has started for breast cancer patients and in breast cancer work with looking at a phage library that tries to find antibodies from patient samples to help direct therapy towards their cells. Did I get that right?
Close enough.
Okay.
I'll fill in the details.
Great, because I'm not going to be able to do the rest of that. He is the S.D. Ireland Professor of Surgical Oncology at the University of Vermont, and as a co-PI with Giselle on a DoD grant to do just this work. And he's going to tell us now all about antibodies and fighting cancer.
Great. Well, thank you. And thanks, Giselle, for calling me. I actually, my mind was in a different time zone. I was in Salt Lake City, and then I thought, oh, no, I got an hour. So, good. Well, the title is can be whatever it wants. Treatment of Childhood Cancers by Amplifying Anti-Cancer Antibodies. And I'll launch right into, what are we going to do? I think, from my perspective, it's pretty simple, because I do basically whatever she says, which I think is true for a lot of people. And that's probably a good thing. So, but the thing that I'll talk about is that we're going to plan on stimulating the immune system. And the way we're going to do that is by vaccinating a patient, and we're going to vaccinate them with their own tumor. And then, once that happens, we're going to boost that response by using laboratory techniques. And why are we going to do that? Well, it's all personal. All this is, the TC is translation, right? So that's where it all has to begin, and that's where it all has to end. So I'm going to talk about, in general, enough background on what is the immune response to a cancer. What's that relationship like between a cancer and the immune system? I'm going to talk about methods to boost that response. I'll talk a little bit about the preliminary data that we've got so far, and then upcoming neuroblastoma studies. So let's kick right in with the immune response to a cancer. So one way to think about this is that why don't all of our cells get attacked by the immune system? And really, they almost do. But there's a fancier word for this called immunoediting, which is what the immunologists use to discuss the impact of the immune system on a growing mass of cells like a cancer. And it starts off with that normal cells have to spend a large part of their time fending off the immune system to convince them that they're okay. Don't eat me. Don't blow me up. Don't hurt me. All right? And the immune system, when you think about it, you think of all the cells that we have in our body. Oh, man. And then think about all the cell parts. That's oh, man. And our immune system has to distinguish between those cell parts, the billions of cell parts, and the billions of cell parts that don't look too much different from other biological things that could hurt us. And it's got to do that in real time every day of our life. This is a phenomenally complex thing. And really the difference between being alive and being dead is an active immune system. You don't live too long with no white blood cells. So let's take a look and see how this thing works. This is a very, very interesting thing. This big oval here represents a cell. Now in the cell there's all kinds of parts. Now some of those parts are made up of proteins, and something very interesting happens with those proteins. This little gizmo right here is a garbage disposal unit, and it's crunching up the proteins, and it breaks them into their little parts, and when they're little parts we call them peptides. Now what happens to those little peptides is they kind of get skittered up in little pieces up to this surface of the cell. They were on the inside, chunk, 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 And now they're on the outside. And they're being displayed by this very special molecule. Now that very special molecule is holding that interior body part and presents it to the immune system. And if it's normal, that's a normal chunk of a normal protein, that cell gets a pass. And it's T cells that come sniffing around to check this out. So let's pause and think about this. This is a pretty amazing thing that the inside of a cell is constantly presenting itself on the outside of a cell and saying, don't hurt me, don't hurt me, don't hurt me. Let's look a little closer at that interaction. So this is that little piece that's being held in the cell membrane, and there's the little chunk of garbage disposed protein sitting there, and here comes the cell, the immune cell, the T cell, and it actually has a piece to grab on and hold and dock for a moment while this piece kind of sniffs it out. And if it says you're okay, it moves on. If it doesn't, well, there's T cells on a cancer cell. They think something's going wrong. A little bit different cartoon, but it's the same thing. Here's a tumor cell or an infected cell. And here's that little piece that holds and displays it, and the little round thing is the displayed chunk of protein. And in this case, the T cell says, that's not normal. I'm going to take you down. And it does that by a series of things. It punches holes in the cell with things called perforins, which is a nice term, perforate. And then once those holes are punched into the cell, the inside can leak out and things can leak in. It blows up, and that cell is dead. And what's interesting about the power of these really very, very potent T cells are that once they're done with that cell, they move on to another one, and they kill that one, and that one, and that one. And they'll do 100. In a tissue culture, you can see them. They're like mowing a lawn. It's a phenomenal thing. So T cells are very, very potent. Now that we've just talked about T cells, let's talk about the B cell story. B cells are the cells that make antibodies. And antibodies are released from B cells, and they circulate about in the blood. Now, before they circulate, they tend to hang out on the surface membrane of a B cell, so we're going to talk about two different things, the released antibodies and also the same antibodies pre-release coating the surface of a cell. Now, the story starts in a lymph node. Lymph nodes are in our armpit, our groin, our neck, our gut area, and they're about the size of a fat pea or maybe a lima bean, and they have these tubes right here and different areas that bring the tissue fluid in that percolates through those lymph nodes. Now, along in that fluid comes what? If that's draining a cancer, then cancer parts are going to float in and enter into that lymph node. If it's a bite from an infection, it's going to percolate in and go into that lymph node. Now, inside that lymph node are all kinds of structural organizations of immune cells. This is a college of immune cells. And they start out in kindergarten. Now, this lower slide shows red cells, which are B cells, and the green ones are T cells. And the point about this slide is that they're in a nice, tidy organization. They really do hang out in their nice areas. But when something comes in one of those pipes, it's a foreign material, and in this slide that is represented by this green fluorescent debris, suddenly those relationships are over. The B cells quickly shuttle up, they grab some of that stuff that's foreign, they run over to the T cells, and they go, how did I do? Now, let's think about a baby B cell. It's got a little baby antibody. And that antibody is a structural shape that's designed to dock on things. Now, in that foreign material, if it comes up and it grabs it nicely or even kind of okay and it runs over to the T cell is very pleased with that and says, good, you get to go over there. If that baby B cell has an antibody that doesn't do that, it comes over and it's got an empty thing, it says, out the door. You don't graduate. Now the graduating class from kindergarten is told to grow and multiply. But they do something else that's really very amazing, and that is they mutate. And they don't mutate everywhere because we know mutations are bad. They mutate just in the genetic code for that antibody. And it scrambles it. And so the kids of that next generation now have antibodies that are all a little bit different. Well, what do? They go running back to that foreign stuff and do the same thing. And the ones that mutated to have a little bit better grab bring more material to the T cells, and the T cells give them a pass and they go to the first grade. And the other ones, out the door. And this goes over and over and over again very rapidly until the graduating class has antibodies that do what? They bind really, really well to that foreign crud, and then they get to go out the door and they get to do their thing. And what their thing is to make antibodies. And then they release them. And this is a cross-section of an electron microscopic picture of a B cell. And this is the part of the cell that makes these antibodies. This is an antibody factory we're looking at right here. And we're going to take advantage of that. So how do B cells do their thing? Well, they circulate around in the blood. You've heard about antibody titers. If the titer's up or down, if you had a tetanus shot, maybe you need another tetanus shot, but they're circulating in the blood. Now imagine one of those antibodies has been educated to bind to a tumor part, and it's circulating along and it bumps into a tumor. It'll stick, because that's what it was graduated and educated to do. Once it sticks and you get a bunch of those, they tell this guy to come over and do something. And what they tell it to do is to kill the cell. And it does it quite effectively. And we heard about NK, the power of NK cells doing that. And they do it very similar to the picture I showed with the T cells, poking holes in it and doing that sort of a thing. So how do antibodies form and to the self, to the body? We could look at a growing cancer as being an autoantibody, a self-antibody. Well, it turns out that autoantibodies, self-antibodies, are really quite common. All right? If I checked almost everybody's blood in this audience, somebody would have levels of antibodies low to almost every major part of your body, but they're low titer. But what happens when the titer kicks up? Then we get diseases. Like, what do you think's being attacked here? What do those antibodies bind to? Joints. Right? It's real. What about the thyroid? Autoimmune thyroiditis. This one here bound to one of the multiple layers of skin, and it just caused sloughing of the layers of skin. In this diagram, it shows the body parts that can be affected when the titers get too high. And the list is really long. You know, you name it, it's going to happen. Which of course makes sense, because it's constantly, every day, while we're sitting here eating, you know, digesting our lunch, our immune system's having to figure out which part is you and which part isn't you. It's going to make mistakes. But we're going to take advantage of this. So let's now switch from the autoantibody to normal things to autoantibody against a cancer. And this is a fascinating study that helps describe this. Took an immunodeficient mouse, and elicited a cancer by injecting a really nasty chemical. And that created a tumor on this mouse. Now this is a mouse with no effective immune system. And so when that tumor grew, a little chunk was taken off and transplanted to another of the same kind of mice. It doesn't have much of an immune system. And it grew. And it grew in that mouse, the next mouse, and so on. It took all the time. What happened when that piece of tumor was given to a scruffy mouse that was a wild-type mouse you might catch in your garage somewhere? That thing's got an immune system that's now attacking that tumor, and most of the tumors didn't grow. So you can imagine all these attacks with all these antibodies and all this stuff killing different parts of the cell. But lo and behold, a minority, 20%, did grow. Those tumors that grew are very different now than the ones that grew in the immunodeficient mice because they've been scrubbed down by an immune system. And this myriad of different cells, many have been killed, but those that could withstand that immune onslaught grew. The ones that grew in the wild scruffy mice, when you took those and implanted them in the next scruffy mice, what do you think happened? They grew just fine. They are now resistant to that particular immune system. So the changing of that tumor from growing in immunodeficient mice to getting scrubbed down, a lot of the cells killed, to now being able to grow in an immunocompetent mouse is very different. It's a very different tumor. It's been sculpted, and that's why they call it immunoediting. I'll give another example. Okay? This is human cancer on the left and human cancer on the right, and all the brown cells were stained with one of the tumor parts called EGFR3, and it's loaded with EGFR3 positive cells. That patient was vaccinated with EGFR, and it mounted an immune response which killed EGFR3 cells. But then the tumor regrew. Now all the EGFR3 is gone, it worked, but it changed that tumor to not have that anymore, but it's still a tumor. Okay? I want you to hold on to that idea. It's an important idea. So let's go back to those mice that had the chemically induced cancer. This circle was a study on one tumor from one mouse to look at all the different mutations that occurred in that growing tumor. There were a couple of thousand. Here's another mouse. It had a same kind of chemical induced thing that had a couple of thousand mutations. There was some overlap, but what was it in the cancer with all these mutations that was recognized by the immune system in the scruffy mice? All right? It was whatever was different. This is my reminder here of the cell and the garbage disposal crunching up pieces of proteins and holding them out on the cell. Here's 2,000 mutations in proteins. They're going to get crunched and moved out to the surface of the cell, and there's lots and lots and lots and lots of targets for the immune system to say that's different. And we're going to take advantage of that. Let's look at neuroblastoma cells. They're tremendously different. Look at the shape of this one. It's all kind of spread out. This is kind of stellate. This is growing in clumps. All right. Here's one patient's cells tested against these tumor part markers, Nestin, GD2. It's got those things, but the other patient doesn't have any of those things. We even see variation, not just patient to patient, but within one patient's tumor, variation. Tremendous, and it would make sense because in this mouse tumor, how many different mutations? A couple of thousand? You know, humans aren't going to be any different. So I look at it like this, right? Colorful M&Ms. Every one of those represents a tumor part. Now, if I kill with my drugs or whatever, my immune therapies, the red ones, the orange ones, the brown ones, and so on, oops, what did I miss? Tumor's going to grow back with all the yellow ones. And all the family members here who have gone for one therapy after another are expert at knowing this process. So the treatment needs to be polyclonal. Poly means many. And that's really going to be the name of the game. And that's what we're going to take advantage of. So how do you create many? Well, one way is by analyzing those mutations using genomic approach. And it's a very powerful thing. This is molecular guided therapy. Okay? Very, very important thing. The other way is to take advantage of the patient's own antibody response to all these different targets. It's not enough to have a response. The titers are low. We have to figure out how to boost that response. If I step back and look at the different types of immune therapies that exist, we have monoclonal antibodies. And are they polyclonal? No. Can they boost the dose? Yes. The pharmaceutical companies that make these antibodies, they get them up very high dose, and you know exactly what milligram to achieve to get some therapy. But they're monoclonal. Tumors that regrow will be resistant to that. Cancer vaccines, they elicit a polyclonal response. But the problem with the vaccines are that you can't really boost it. You give a vaccine, you get a response, you get what you get. Well, then there's T-cell therapy, and I'll show one slide on this, and NK therapy may be very similar to this sort of approach, but basically that's polyclonal, and the response is boosted. Now, with antibodies, it's really never been done to get a polyclonal response and to really control the level of antibodies that are achieved from that response. And that's our plan, and I'll show more details about this. But what do you get when you do achieve polyclonality and boost the response? This is data from Steven Rosenberg at NCI, where he took tumor-infiltrating lymphocytes, these are T cells, from a tumor. In this case, it was melanoma patients. And then he got those purified, and he amplified them in the lab. They're polyclonal, they target lots of different things, but when he gave those back, he now has a high dose. And he got up to 40% complete response. Complete response meaning the tumor is gone. These responses were rapid. They occurred in 2 to 4 weeks. And I think the really very incredible part about this is that these were durable. These data go out now to a decade. And so essentially we can talk about these patients being cured. The problem with this is it's very difficult to do, and very few patients are actually eligible to get this. But in those patients that this has been done, it's been extremely effective. And I think it's because it's polyclonal and it's boosted. That's the theme. So why are we shifting over to polyclonal antibodies? Well, T cells, even though they can be extremely effective, they're only part of the immune response. We have the T cells and we have the B cells secreting antibodies, and they work harmoniously together. So T cells are only part of this. What is the other rationale to approach neuroblastoma protocols this way? In animals, vaccines can induce antibodies that can eradicate established tumors. And clinical trials that have been done with vaccines, even though I already mentioned that you can't control the antibody level in any given vaccine patients, if you check the titers, which many people do, you can correlate that very directly with the response, meaning that the higher the antibody level, the better the response. It's very clear. It's the dose-response relationship, which is no surprise. The other thing is multiple antibodies are better. It just is. It's experimentally shown, and now clinically with antibodies that target adult cancers. When they use 2 of them, they get a better response, even just 2. But there are no reports on a controlled method to increase the blood levels of vaccine-induced antibodies. And it was very surprising to me that this was such a blank area of immune therapy. Well, let's talk a little bit about what we've got so far. So where should we get antibodies? Well, we could get them from the blood, but the blood's just got everything we've ever been exposed to circulating around to the last virus we were infected with. But a tumor drains, as I mentioned, through the lymphatic system to these lymph nodes. These are called sentinel nodes if it's a cancer. But imagine that's injecting a vaccine right there. Where's that vaccine going to go? It's going to go to that lymph node right there, and we have technology now using tracers that we inject around the skin that get absorbed in that lymphatic and make that lymph node very easy for me to identify as a surgeon. And it's done with a probe a little bit bigger than this pen. And so if I inject a vaccine right here in the thigh, just like that picture, it's going to drain up. And I can find that, and it pegs it right to a precise spot. And I can remove that lymph node under local anesthesia through a tiny 1-inch incision. Now, what's in that lymph node? All those B cells that have gone through college, and they're ready to go, and we're going to boost them. Now, the other place that lymph nodes live are in the marrow, and so that's another place we can look, and this gets to some of the data I'll show in a moment that we've gotten from Giselle's patients. But we've made human antibodies from tumor-draining lymph nodes. And so how do we do that? Briefly, we grow the B cells in the lab. And unlike NK cells and unlike T cells, B cells do not like growing. They just peter right off. But we grew them transiently. They secreted antibodies, and we took those antibodies and tested them. Every one of these spots is one of the little clones of antibodies from different B cells. And then we layer over that crushed up tumor. And if one of those antibodies binds to a tumor part, then that crushed up tumor part will stick. And that's what we see right in that spot. Fantastic. So we went running back to that little well that had that one clone of B cells, and they were all dead. Fine. Okay, that sucks. We're so serious. So now we go, and now we have to do something different. You have to grow them for a long time. Now, in order to do that, it's complicated. It's very challenging to do, and what you have to do, I won't go into a lot of detail, you have to take the B cell, which doesn't grow, but it makes antibodies, and you take another cell that grows really well, but it doesn't make antibodies, and you wham them together. You make a hybrid. We call it a hybridoma. And if those two cells, when they're smushed together, form one cell, can survive that, most of them don't, occasionally one of those new cells will do what we want, which is to make antibodies and grow. And you do that by whamming them together and then giving them an electric shock just below blow-up level. So they kind of fray out like this, and then they fuse back together. And if they're immediately adjacent to another cell, sometimes they just whoop, and they come together. You can see this happen under a microscope. It's very cool. And that's these two cells here that have fused, and now they form one cell. It's very inefficient. You get 1 out of 100,000 successfully doing this, but it turns out that a lymph node, just a normal lymph node, has about 500 million B cells. So even at 1 out of 100,000, you're still going to get thousands of successful hybridomas. And it works. It really works. All these are anti-tumor antibodies we got from a patient with breast cancer from those nodes, and it binds to all these different tumor cell lines. The important thing is that once you've got that antibody in hand and you've got it growing and actively secreting, you got it. And now you can do all kinds of things with it. You can test it, you can replicate it, you can put it in animals, you can find out what it's binding to. So this little chart here is a smear of a tumor cell, and then it shows binding of the antibody to that little smeared out tumor cell part. I don't know what it is yet, but it's going to be interesting. And I know that if we hadn't have this growing clone, we could never test anything like this. So we've done this on the cases from Giselle, doing it on bone marrow. And so these are the first time we've taken neuroblastoma cancer cells and the B cells isolated from the bone marrow from one person and converted those B cells into these actively growing ones and then tested them back. And so these are neuroblastoma cells from the same patient from which the B cells were derived, which is extremely cool, and it shows binding. And you always, all the doctors have to show the things that don't work. So I could have just as well put that as a blank slide, but there really are cells there. but they didn't take up. So that's what a negative looks like. This is one of the same tumor that grew in one of those immunodeficient mice. So now we have a whole cluster of growing cells. And that antibody from that patient binds not only to their cells growing in culture, but it binds to the tumor growing in the mouse. So what we're looking at is really more or less an approach, a universal platform, whether it's neuroblastoma or glioblastoma or breast cancer. Just like the immune system can go after body parts, it can just as well go after cancer parts. And we're going to take advantage of that. And as we move to clinical trials, we've got to speed all these things up. And so we worked from these little wells, these little plates, instead of individual test tubes, we go to 96. That speeds things up almost 100-fold. We can go up to 384, and now we've got a robot that can spot those little things, and we can go up to 10,000, although that's pushing it. But even thousands is phenomenal. We're also working with these amazing engineers at MIT who have micro-etched things that will actually hold one cell at a time. And this thing, the size of a glass slide, has 80,000 wells etched into it. We're going to put B cells in those wells, and maybe we can ramp up our screening time, 1,000-fold. So for switching to the end of really talking about proposed neuroblastoma study, I want to kind of remind, why is it important to be polyclonal? Well, there's lots of different targets of antibodies, not just kind of the parts I've described, but people know very clearly that antibodies will bind to carbohydrates. Those are different parts. They'll bind to circulating growth factors, gangliosides, really all the different major categories of cell parts antibodies bind very nicely to, thank you. The other thing is that antibodies don't work just by one mechanism. They work by lots and lots of different mechanisms. One way is ADCC, antibody-dependent cell cytotoxicity, and that's where it tells another immune cell to come kill. But it does a lot of direct things. Inhibits cell migration, fixes complement, directly inhibits various proteins. It's just a real protean system. We've got a protocol that we started in collaboration with Dartmouth. We got through the FDA, which is a very tedious thing to do. And in that case, we're taking a vaccine. We're going to take the tumor, smush it up, and make what's called a lysate, and inject that into the thigh of the patient. Do that twice. A few weeks after that, I'll pull out that lymph node, and from that lymph node, we will generate our antibodies. And that's open for accrual as of 2 weeks ago. For a neuroblastoma study, same idea. Whether it's a tumor specimen that's obtained surgically, or whether it's the filtered ones from the marrow, which would be extremely cool, we'll generate vaccines from the patient's own tumor. And then vaccinate the skin. That's what this little diagram is here, injection site. That will go up to a node that I know I can find very confidently, and I can remove it safely under local anesthesia. After we get that, we immortalize the B cells. We screen those for tumor binding. And after that, we deliver those amplified antibodies. And like you've heard in all the really good presentation this morning, you start off going for safety and then look for efficacy with increasing doses of these boosted anti-tumor antibodies. So our goals really are to develop an entirely new method of gaining what I call command and pharmaceutical control. When you give a vaccine, you know you got all this stuff going on, but it doesn't get boosted. It's got to be boosted. And we have a way of boosting that response to really clear-cut levels. You want to get up to so many milligrams, you grow more. You can go to any level that you want. Now, I want to also kind of share with you the conceptual part of this thing. So my little guy represents polyclonality, right? There's the picture. If you don't kill all the red ones, you're going to have it. Whether it's a stem cell, which you heard this morning talking about, you got to get them too. They're probably the green ones, I think, the stem cells are the green ones. And you boost that thing. Now, even though I'm a tremendous optimist, I know that after we shrink tumors, that some of those are going to regrow. And if that was the end of the story, I'd be very disappointed. What this does is allow us to have a process rather than a single therapy. So when that tumor regrew, what would it be like? Would it be the same tumor as before? No, it's going to be a different tumor. Well, we'll take that tumor, we'll do this thing all over again. Throw that at the immune system. And so that's what the little bicycle racer is. You do it again in quick order. So we're really trying to develop a process. Well, that was my last talk. And I know when I go home, you know, you sit around with your family and describe, oh, and I saw Giselle. It was great to see Giselle, and I met some new people, and so on. But you know, I'm going to tell them that I had a moment with this very sweet little girl stuffing her face with cake, and that's really one of the big highlights for me at this meeting. So thank you very much.